Dear learners, in my last lecture, I discussed Calvin cycle, which has three steps: carboxylation, reduction, and regeneration. In first step, that is carboxylation, carbon dioxide is hooked to a 5-carbon sugar RUPP and producing two molecules of 3-carbon acid or 3-phosphoglyceric acid. And this reaction is catalyzed by an enzyme called as Rubisco. Rubisco is abbreviation of ribulose 1,5-bisphosphate carboxylase oxygenase. So somebody did a great service to humanity by abbreviating it as Rubisco. So this Rubisco or ribulose 1,5-bisphosphate carboxylase oxygenase enzyme fixes carbon dioxide in Calvin cycle. So it acts as carboxylase. But the problem with this Rubisco is that it can also fix oxygen instead of carbon dioxide. So Rubisco is like a good friend with a bad habit. So in this lecture we will discuss what happens when Rubisco acts as oxygenase. So here we have Rubisco. When it accepts carbon dioxide, it gives two molecules of 3-carbon acid or 3-phosphoglycerol. So this is a perfectly okay reaction. But when it acts as oxygenase, fixing oxygen instead of carbon dioxide, it gives one molecule of 3-PGA and one molecule of phosphoglycolate. So 3-PGA is okay, the production of 3-PGA, this happens in the normal photosynthetic carbon reduction cycle. But this compound, glycolate, this is problematic. This is a toxic compound, and plant has developed an intricate mechanism involving a series of metabolic reactions in 3 different organelles to get rid of this glycolate. And this phenomena is termed as photorespiration. So Rubisco is a bifunctional enzyme which can act as carboxylase when it accepts carbon dioxide, or it can act as oxygenase when it acts as oxygen. So when it acts as oxygenase, then it produces one molecule of phosphoglycerate and one molecule of glycolate. So how does plant get rid of this glycolate is the topic of our discussion today. what is called as photorespiration. But before I start discussing how plant gets rid of glycolate, I would like to talk about the properties of this rubisco. Rubisco is the most abundant protein in nature. It makes up 50% of total leaf soluble proteins. So plant invests a huge amount of its nitrogen into this rubisco. So why is it so? Because it is a large protein. Rubisco is a large protein with a size of about 550 kilodalton. So it consists of 8 large subunits and 8 small subunits. Large subunits with a size of 50 to 55 kilodalton and small subunits with a size of 13 to 18 kilodalton. The genes of large subunits are encoded in chloroplast, whereas that of small subunits in the nucleus. So this is a large protein. In stroma, its concentration is about 4 millimolar. Not only is the size of this protein is large, but it is also a highly inefficient enzyme. Inefficient, sluggish enzyme. It can fix 3 to 10 carbon dioxide molecules per second. And this catalytic activity is too slow. For active enzymes, the catalytic activity is in the order of thousands or millions. So this is such a sluggish enzyme. And even it requires another catalytic protection enzyme called as rubisco activase, which is a chaperone for its own activation. And then it has poor specificity. As I told you before, that it can bind or it can react with both carbon dioxide and oxygen. So carbon dioxide and oxygen are competitive inhibitors. They bind at the same site. This is probably due to the fact that rubisco evolved at a time when the atmosphere was devoid of oxygen. So, and with the development of oxygenic photosynthesis and increase in the concentration of oxygen in the atmosphere, rubisco has become a victim of its own virtues. So now for every 3 molecules of carbon dioxide fixed, 1 molecule of oxygen is also taken up by rubisco. So the ratio of photorespiration is about 30 to 40% in C3 plants. So rubisco is a sluggish and inefficient enzyme with very low catalytic activity. It has poor specificity for carbon dioxide and oxygen, and it is a large protein. So for these reasons, plant has to invest a huge amount of nitrogen into this rubisco, and this equates to about 10 kg per person, the nitrogen investment in Rubisco. So in C3 plants, as the concentration of oxygen increases, the rate of photosynthesis decreases. So it is a negative linear relationship. And Rubisco is more active under low temperature conditions. So as the temperature increases, Rubisco activity decreases. Although the affinity of Rubisco is 80 times more for carbon dioxide than oxygen, but the concentration of oxygen in our atmosphere is 25 times greater than carbon dioxide. Oxygen concentration in the environment is about 21%, whereas carbon dioxide concentration is just 0.039% or 0.04%. With the increase in temperature, the solubility of gases in water declines, but the solubility of carbon dioxide declines more rapidly as compared to oxygen. So high oxygen concentration or low carbon dioxide concentration in the atmosphere or high temperature, these are the conditions that favor photorespiration or that favor the reaction when Rubisco acts as oxygenase enzyme. So this phenomena is called as photorespiration commonly, or C2 glycolate cycle, or photorespiratory glycolate pathway, or photosynthetic carbon oxidation cycle. PCO. So there are different names to refer to this phenomenon. So let me write down the complete cycle, that how plants get rid of this toxic glycolate compound. This C2 glycolate cycle involves three organelles: chloroplast, peroxisome, and mitochondria. So in the first step, when Rubisco acts as oxygenase, the first reaction is oxidation reaction, in which two molecules of oxygen combine with two molecules of RUBP to give two molecules of 2-phosphoglycolate and two molecules of 3-phosphoglycerol. This is a 2-carbon compound. So this phosphoglycolate is dephosphorylated to give two molecules of glycolate, 2-carbon compound. So a total of 4 carbons move along this path. This glycolate is then exported to peroxisome, where it is oxidized to glyoxylate and hydrogen peroxidase. The peroxidase is broken down by catalase, whereas this glyoxylate is converted to glycine in a transamination reaction. This glycine is a 2-carbon amino acid. This glycine moves to mitochondria, where it loses 1 carbon dioxide. So there are 2 molecules of this glycine, so a total of 4 carbon. It loses 1 carbon dioxide and is converted to another 3 carbon amino acid, serine. So this is a 3 carbon amino acid. This serine then moves to peroxisome, where it is converted to hydroxypyruvate in a transamination reaction. Hydroxypyruvate is reduced to pyruvate, 3 carbon sugar. Pyruvate then moves to chloroplast, where it is converted to phosphoglyceric acid using 1 ATP, and this phosphoglyceric acid then enters the Calvin cycle or C3 cycle that we discussed last time. As a result of the C3 cycle, RUBP is regenerated, and this 3-PGA can also enter this C3 cycle. So total of 4 carbon moves along this path. 1 carbon dioxide is lost here, down here in mitochondria, and 3 carbon are recovered. So in this way plants get rid of this toxic compound glycolate and recover 3 out of total 4 carbons. But there is some additional cost involved. 2 additional ATP are required for this process. 1 ATP to convert pyruvate to phosphoglyceric acid, and 1 ATP for reassimilation of this ammonia, which is reassimilated by the enzyme of glutamate synthase cycle. This also becomes toxic if this is not reassimilated. So in this way, this process of photorespiration is linked to photosynthesis, as it is regenerating RUBP, and also to nitrogen metabolism. Sometimes photorespiration is confused with mitochondrial or dark respiration, because it also involves the uptake of oxygen and release of carbon dioxide. But this is different from the mitochondrial respiration or dark respiration, because this depends on the process of photosynthesis for the enzymes and for the regeneration of RUBP. So this is not a normal respiratory process. So 2 ATP 2 additional ATP are required, and 1 carbon dioxide that was fixed is also lost in this process. And you learned in the Calvin cycle that to fix 1 molecule of carbon dioxide we need 3 ATP and 2 NADPH. So loss of 1 carbon dioxide also means the loss of 3 ATPs and 2 NADPH. So this is a wasteful process which wastes energy and the already fixed carbon dioxide. And this is found in plants called as C3 plants. So they have developed this intricate mechanism involving 3 different organelles: chloroplast, peroxisome and mitochondria, in which this series of metabolic reactions are performed to get rid of this toxic glycolate. Is there any benefit of this glycolate pathway, or why do plant do photorespiration? One advantage of this photorespiration mechanism is that under conditions of excess light and carbon dioxide limitations, for example, plants have closed their stomata in response to water shortage in their root zone. So now carbon dioxide is limited, but there is bright sunshine and plant is producing ATP and NADPH. This excess energy can cause photooxidative damage or light induced damage, because due to absorption of excess energy, chlorophyll can enter triplet state and it can oxidize the whole photosynthetic apparatus. So one advantage of this photorespiratory pathway is that it acts as additional sink for this wasting this additional absorbed energy by dissipating it in the form of heat, and the ATP and NADPH which are in excess of the plant, which are not being utilized in the carbon reactions, can be dissipated in this way. So naturally plants have evolved some mechanisms that we will discuss in the coming lectures, that is a C4 pathway or glycylation acid mechanism to overcome this photorespiration phenomenon. But there are also efforts are underway to find alternate mechanism to avoid this photorespiration in some of our most productive crops like rice, wheat and soybean, which are most important from nutrition point. So this is all about today's lecture. In photorespiration, visco acts as oxygenase, so it takes up oxygen instead of carbon dioxide, and it produces a toxic compound glycolate. Then in a series of metabolic reaction involving 3 different organelles, plants get rid of this toxic compound glycolate and recover 3 out of 4 carbon. But this incurs additional cost, so thus wasting energy as well as the already fixed carbon dioxide. But the advantage of this photorespiration is that it can prevent or avoid the light induced damage.